Okay. Jill, what do you think? You ready to go? All right, everyone. Good morning, good afternoon, depending on where you're joining us from this morning or this afternoon. Welcome to the 9th session of ARCS Forward and the National Scholar Speaker Series. Thank you for tuning in today. I'm Diana Gordon, President-elect of the ARCS Oregon Chapter, and I will be moderating our session today. I'm a former high school biology teacher who moved out of the classroom in 2004 to run the community education program at the Oregon National Primate Research Center. And I recently retired from that job at the end of January. I joined ARCS Oregon in 2016 because it gives me the opportunity to connect and support two of my great passions, science and students. ARCS stands for Achievement Rewards for College Student Scientists. The mission of ARCS Foundation is to advance science in the United States by providing financial awards to academically outstanding U.S. citizens studying to complete degrees in science, technology, engineering, and medical research. ARCS Foundation has a long history of furthering this mission. Since our founding in 1957 in Los Angeles, ARCS volunteers have been working to elevate young scientists by providing them with unrestricted financial awards. If this mission speaks to you, as it does to me, I encourage you to consider joining us in our work if you're not already a member of ARCS. To learn more, talk with an ARCS member, or visit our website at arcsfoundation.org to find a chapter to connect with in your area. During our ARCS Forward session today, we will be learning about the deep ocean and about an amazing creature that resides there. We have two current scholars doing equally impressive yet different work in this area. I know you will enjoy hearing from Jessica Perelman from ARCS Hawaii and May Songco-Casey from ARCS Oregon. These ARCS Forward sessions have covered a wide range of STEM topics. The ARCS Forward National Scholar Speaker Series was created to inform the audience about science topics that are relevant and interesting to all audiences, whether you consider yourself science-oriented or not, and to introduce scholars from across the ARCS network of 15 chapters to our members, donors, and friends, and to other scholars working in related fields. On behalf of the ARCS Forward Committee, thank you for joining us today. As you listen to the speakers, please feel free to enter questions into the chat. There will be a time for Q&A at the end. Please welcome Jessie Perelman and May Songco-Casey.
All right, Diana, thank you very much for that introduction. I will go ahead and kick it off and share my screen. All right, is everyone able to see this? Great. Well, hello, everyone. As Diana mentioned, my name is Jessie Perelman. I was an ARCS scholar in 2021 during the completion of my PhD at the University of Hawaii. I'm currently now working at the NOAA Pacific Islands Fisheries Science Center through the Cooperative Institute of Marine and Atmospheric Research. And today I'm going to be talking to you all a little bit about the work that was largely supported by the ARCS award that I received during my PhD, and that was understanding the behaviors of deep sea animal populations in the remote open ocean. And specifically, we were focusing on what the implications of some of this research was for a developing deep sea mining industry and changing oceans. So what you see here is just a photograph of some of the sampling tools that were used to conduct this research, and you'll have a bit of a better understanding of what these were used for in just a moment. So as I talk about the deep ocean, I want to talk a little bit about the different animals and creatures that live there, and specifically what part of the ocean that my research focused on. So this beautiful graphic, this was created by the Woods Hole Oceanographic Institution, and this shows different depth zones of the ocean and some of the different animals that live there. So you can see this top zone, or the euphotic or epipelagic zone it's called, is the upper ocean, typically down to about 200 meters. And this is considered the depth where light is able to penetrate down to this depth, and a lot of the animals that live here rely on this light for food availability and ocean productivity and all that. And you might think of some of the apex predators and fish that we eat, like mahi and tuna, especially important here in Hawaii. And then when we get below 200 meters, the downwelling light from the sun starts to disappear, and we get into this area called the twilight zone or the mesopelagic zone. This is roughly characterized by the waters that are about 200 to 1,000 meters in depth. And you can see quite a change in the animal communities that live here from these maybe more typical marine animals that you would think of to these maybe more slightly strange looking or deep ocean dwelling creatures. You see a lot of fish and jellyfish and crustaceans here. And so I just want to point out the importance of all of these different animals to each other, because they are all connected despite these differences in these different depth zones. They're really critical to each other in ocean food webs. So, all of these animals may feed on each other or feed on, prey of another's prey. And so everything is connected both vertically and horizontally in the open ocean. And specifically today, I'm going to talk to you a little bit about this particular group of animals that's collectively we call micronectin. And this is a fancy word that's talking about these animals that are maybe in the middle of the food web. So they're often directly eaten by these top apex predators like these tunas and mahis, but they themselves might eat the smaller prey like little zooplankton or smaller crustaceans in the water column. So micronectin includes the small fish and squids and crustaceans, usually only a couple of centimeters inside, that occupy this middle of the food web space, and they're often not really big, and they're often considered prey communities. And just a really cool graphic here to show you the diversity of some of these animals. As I mentioned, they can look kind of weird and kind of cool, but there's vibrant colors. A lot of these animals might be red. A lot of these animals just might have big eyes, big teeth, these different adaptations to living in deeper, darker waters. And I've named some of the fish of interest and animals of interest that we're focused on in some of this work. So different fishes and gelatinous animals like siphonophores and pyrosomes that you may be less familiar with. And one of the really, really cool phenomena that a lot of these micronectin or midwater animals participate in is something called daily vertical migration. And this is another beautiful graphic that I think really does a good job showing this phenomenon. So a lot of these animals will reside down at deeper depths during the day in those darker waters to hide out from those top apex predators during daylight hours. And then as the sun goes down, many of these animals will migrate up to the surface, as you can see here in this graphic, to go feed in those more productive surface waters where there's food availability at night when it's dark and are less likely to be seen by those apex predators. And then when the sun comes up again, they will migrate back down to that depth and hang out there during the day. So not every single animal does this, but a good amount of them do. And if it's any insight from this graphic, these organisms are really, really abundant in the open ocean. There are probably trillions and trillions of these animals. And what you can see here is that there's some important processes that this vertical migration plays a part in. And one of those is called the biological pump. So as these animals migrate up and down in the ocean, they're transporting carbon and nutrients from the surface where they're eating down to the deep ocean where they might excrete that food and respire. So again, this is a unique phenomenon and part of what was a huge focus of the research that I'm going to talk to you about. So I'm talking about the open ocean, and I want to point out what part of the open ocean that I'm referring to. So a lot of my research focused on this area east of Hawaii, west of Mexico. I'm going to generally be referring to this as the Eastern Pacific Ocean, and I've boxed it here so you get a general sense of what area I'm talking about. And one of the main reasons for the focus on this area, as I implied during the introduction, is that it's currently being targeted for a developing mining industry. This is deep sea polymetallic nodule mining. So I'll just show another graphic here that currently shows what the infrastructure is starting to look like for this mining industry. So these black polygons that I just put up on the screen show the current licensed mining exploration areas. These are each sort of designated to different countries and different mining contractors, and they're governed by a UN body called the International Seabed Authority. And so this is, again, a developing industry, and these various countries and contractors are going out and exploring the potential to mine these nodules that are full of very useful metals for things like our electric car batteries and computers and electronic devices across this huge swath of the Eastern Pacific Ocean. And these gray boxes you see represent no mining zones. These are meant to be marine reserves where no mining is able to occur in order to have some sort of protections against the potential detrimental effects of this industry. And as you can imagine, there's quite a few detrimental effects that we might expect from this developing industry. So this is a graphic from a recent paper in 2020 that just shows what an expected mining operation is likely to look like. So at the top, you see this vessel at the surface. This is the mining vessel, and it has this this very, very long riser pipe extending all the way to the seafloor. And if you're wondering how deep that is, that's several miles under the seafloor, or under the sea surface, excuse me. The seafloor here is about 5,000 meters or 5 kilometers. And the general idea is that there's this mining collector machine that drives along the seafloor and just collects these nodules that are sitting on the seafloor and sends them up through this riser pipe. Oops, where did my cursor go? All the way back up to the sea surface. And you can imagine this is likely to create a lot of noise. There's a lot of expected sediment plumes from this collector vehicle driving along the bottom. And not only that, but then once the nodules are at the surface, then they need to send back down that discharge water that's full of sediment and little particulates from those nodules down back into the water column. This is called the dewatering plume. And this can likely spread a very, very expansive distance. And so this is likely to have strong influences on that midwater area that I was talking about, all of these unique organisms living in. So this midwater habitat. And so this, as I've circled here in red, is a big part of the reason that we wanted to focus on studying this particular area and studying some of the animals that live there to be able to create a baseline understanding of what's going on in this huge region that's suddenly being targeted for this this developing industry that could be quite substantial. And we really don't know much of anything about this region or the animals that live there. So in order to go out to this very remote part of the ocean, as you can imagine, it's quite difficult to get out there. It can take days and days on ships just to get to either end of the area. So we try to use a variety of different sampling tools to go out and sample the different biology and chemistry and physics of this area. And as I mentioned, we're focusing on this micronectin community of animals in the middle of the food web. So one of the main ways that I have done this in my research is using something called active acoustics. And so this graphic here on the left tries to do a bit of a good job to explain what that means. So if you can imagine, you have a ship at the surface that might be doing whatever it's doing, and it has a sonar system you can think of that's sending these sound waves down into the water column. And as those sound waves reflect or hit the different animals that are in the water column, that sound then reflects back to the ship and lets us know where those animals are and how they might be behaving. And so when we see aggregation of these animals together and the sound is reflecting back, we refer to these as scattering layers. So we'll have deep scattering layers that we can see migrating down and up during the daytime and nighttime from the sonar imagery. And then we can also see layers that maybe don't migrate at all. They might be a bit deeper. So you'll hear me refer to scattering layers here, and this is just a proxy for these animal communities when we look at them with this active acoustics technique. But there's several other ways that we study these animals as well, and I use these in my research, but I just wanted to highlight them here as I won't focus on them too much in this talk. So one of the other very common ways to collect animals is by trawling nets through the water column. And so this image in the middle shows an example of just how massive these nets can be to try to catch these tiny little critters, because you can imagine it's a big part of the ocean that you're trying to sample. And so it takes quite a team of people to haul these nets in and out of the water. It's quite a big operation, but another complementary way to sample these communities. And then finally, video surveys are also very, very helpful, especially for animals that might be really fast swimmers, and they can avoid being captured by those nets. So this here on the right shows an example of effectively a robot. We call these remotely operated vehicles that are able to be controlled from a ship at the surface and go down to deep depths in the ocean and take videos of different animals that live there. So for the rest of this discussion, I'm going to focus on the active acoustics component and hopefully give you a bit of insight into what we were able to discover by studying this part of the ocean. So I just want to show you what those sonar imagery, or the we call them echograms, look like when we actually take images of these sound waves reflecting off animals in the water. So on the right here, this is an echogram. The colors represent the amount of backscatter in the water column. And so higher backscatter or stronger scattering layers, I might say, represent higher densities of animals, or just animals that might have a stronger signal from that acoustic sound wave. And then cooler colors represent the fact that there's less in the water column, or the animals there might not have as strong as a signal. And this shows 3 cycles of daily vertical migration. So you can see that day-night pattern of animals migrating down from the surface at night, or at dawn, and then hanging out during the day and migrating back to the surface at night. And another way that we can look at this, this is just another way to look at a single daily vertical migration of these animals. You can just see that they follow that same migration pattern. But one other way we can look at this is as just a vertical profile. So I just wanted to show this as an introduction to another figure that I have. But here we see on the y-axis that we have that same depth, and on the x-axis we see that backscatter strength. So this is another way of looking at those scattering layers, where during the day we see that it's this strong scattering layer that's down just above 400 meters, and then at night we can see it migrates up to the surface. And so this is just different examples of ways that we can look at the strength of this backscatter, how pronounced or broad these scattering layers are, and what that might represent in terms of the animal community there. So the research questions that I wanted to address specifically throughout my dissertation and the work that was supported by ARCS was primarily what ocean features might influence this vertical migration behavior, as well as the distributions of micronectin across this Eastern Pacific Ocean area. And from this, I wanted to generally understand what are the potential effects of a changing environment on these deep sea communities. And this changing environment we expect to occur from activities such as deep sea mining, but also just general changes potentially related to climate change and other human activities. And finally, I wanted to understand what do these different sampling approaches that I discussed a minute ago reveal about the different ways that we might perceive these communities, or how we understand them, because we might be seeing different components of these communities as we look at them with different lenses. So I just want to take a few minutes to talk about this first research question for the sake of time, and highlight some of the really key findings that we had in going out to this really remote region and studying these animals there. So when I talk about ocean features, these can be really, really big features or very small features. And so we tried to focus a little bit on both of these spatial ranges. And one of the really prominent features in the Eastern Pacific Ocean that I want to bring up is called an oxygen minimum zone. And this is an area where those midwater depths are really, really deficient in oxygen. So you can imagine, just like we do, these animals need oxygen in order to survive. And so the level of oxygen present in the water column can vary at different depths in the water. So this here shows a map of that Eastern Pacific Ocean. We've got latitude on the Y and longitude on the x-axis. And these different contours show just the amount of oxygen that's present in that midwater depth between 200 and 1,000 meters, that twilight zone. And so the warmer colors represent more oxygen, and the cooler colors represent less oxygen. And what you see here is this really clear, I would say, tongue of low oxygen coming from east to west across this region, indicating that the core component of this Eastern Pacific region is really, really low in oxygen. And so we can expect that this would influence the animals that live there. And sure enough, when you look at some of those acoustic echograms that we've collected, we see really strong differences in the backscatter characteristics, or these scattering layers, in areas with high oxygen, like you can see here on the left. We have, a lot weaker layers. We have some non-migratory layers, versus on the right, this is a lower oxygen area. And we see that these scattering layers are stronger and much shallower, and pretty much everything is migrating up. They need to get that oxygen at night, so they can't stay down in the deep water column where there's no oxygen. So this is one of the key findings that we found, is that there's a strong difference in the way that these animal communities behave depending on oxygen availability there. And now I want to just briefly talk about a smaller scale ocean feature that we also observed to be very important. So that oxygen component was looking largely at the whole Eastern Pacific Ocean, but another more ephemeral feature that is very common in this region are called eddies. And so an eddy is effectively a rotating body of water. It can be spinning clockwise or counterclockwise, and this determines sort of what other influences that this feature might have. So for instance, a clockwise spinning eddy, these are propagating from east to west across the Pacific Ocean. These might cause the water to come up to the surface or to be pulled down from the surface, and then this can influence the way animals interact with it. So you can imagine that potentially these small animals could get entrained in one of these spinning bodies of water, or they could be, moving up or down in the water column with some of the effects of these eddies are having on them as they propagate across this region. And this map here shows what some of these features look like in the way that we study them. So this is showing a sea surface height, and sea surface height lets us know what type of feature we're seeing. So this would indicate to us that these are eddies that are rotating clockwise, and so they're pulling water down into the water column. And this might bring warmer waters down to deeper depths that are usually cold. And one of the really cool observations that we had from this is that these features seem to influence the scattering layers that we observe from active acoustics. So on the left here, this is an example showing some survey tracks from a vessel that we had go collect acoustic data, and the different colors represent different groups of different types of layers that we observe. So the more similar scattering layers that we found, we grouped them together to see how they might be different and what might be driving that. And this plot here on the right shows those scattering layers, again, with the depth on the y-axis and the backscatter strength on the x-axis. And we can see that there's quite a difference in the structure of these scattering layers. So two of the layers, the blue and the red, are quite a bit shallower and stronger than the other scattering layers that we observed when we sampled just southeast of Hawaii. And really interestingly, we found this to be corresponding to these different eddy features that were present in the area during the time. So these are actually spinning counterclockwise. So these are bringing water up to the surface, bringing cooler waters up from depth, potentially enhancing productivity and food availability for these animals, allowing them to reside at shallower depths and perhaps feed in shallower waters. So in addition to really big features like that oxygen minimum zone, we can see that these smaller features that are more ephemeral and they might just last a few months also really have a huge impact on the behaviors of these micronectin communities. And as I mentioned, these eddies are very, very common across the Eastern Pacific during certain times of the year. And so this could have implications for the mining industry and understanding what behaviors in these animals might be driven from natural variability, like these eddies coming through, versus what changes might be occurring due to influences from the mining practices itself. So just to drive home some takeaway points from all this research, the main findings that we had were that deep sea communities are very dynamic in space and time. So they're not static, they move around a lot and they behave differently. And these ocean features that I mentioned play a really important role in driving this natural behavior variability amongst these animals. And this variability is normal, it's inherent in the open ocean, and it plays a really large role in things like animal aggregations, predator-prey dynamics. Some of these features like eddies are targets for commercial fisheries because they're known to enhance the abundance of some of these animals. So the variability that we see from these features is very important and a normal feature of open ocean habitats. And then finally, understanding this variability and how these these different features interact with animal communities in the open oceans is going to be helpful in distinguishing differences between what is natural ecosystem changes occurring from these features versus what might be changes that are occurring that are induced by human activities like mining in the future. And I just want to say a big thank you to all of the collaborators in this effort. You can see it took a massive team of people to go out and into the middle of the ocean several times and collect all this data, as well as various funding sources, including ARCS. And so I really couldn't have completed all of this research without this huge team of support. With that, I will throw this up for questions, but I think we're going to do questions at the end. So for now, I will stop sharing and turn it over to Mia.
Thank you, Jessie. That was an incredible amount of information that you made very clear. It's very clear how interwoven and interconnected everything is, and how complicated that makes everything. Now we'll hear from May.
Okay, can I get confirmation that I'm sharing the right screen and it's not presenter view?
This looks great so far.
Okay, perfect. So before talking about my research, I first just wanted to say thank you to the ARCS Foundation, both for inviting me out to share about my work, but also for the impact that it's had on my research and my personal development as well. I wanted to highlight the ways that ARCS has impacted my professional development and personal growth through some ways that I initially did not expect. So as a since the ARCS Foundation provides unrestricted financial resources, initially beginning grad school, moving away from my family, and being the first in my family to pursue a PhD, it was very isolating even without a pandemic. The ARCS Award came in by allowing me to connect with family and build a community with friends where I was able to pursue activities outside of research. It also helped me connect both with my partner, myself, and nature. So I'm really grateful that I was able to use these funds to start building a community in this new place, but also start building a savings, which was extremely important, especially in the last year. What many of you may or may not know is that when conferences come up, grad students typically have to put forward those costs before they can be reimbursed, and sometimes that can take months. So in some cases, I've had to turn down opportunities to pursue these professional development opportunities, but in other cases, I was able to use the savings that I had to attend conferences both within the US and outside of the US. By knowing that I had financial support to be able to pursue these, I spent less time worrying about how much is it going to cost, like what dent is it going to put in my wallet and stuff, and I could focus more on what I would be able to get out of attending these conferences. But what I think was even more impactful from the ARCS Award is the fact that I had this unlimited community support. By having all of these people believe in me and my ability as a scientist, I had the confidence to kind of pursue science communication outside of the university. And I also put together a video to help encourage Congress to continue funding science because I was inspired by the message of the ARCS Foundation. And recently I gave a talk on the East Coast at Rutgers University, sharing about my work there. So I just wanted to thank you all for believing in me even before knowing about my science. But since I'm here to talk about my science, I'm really excited that I get to share the work that I've been doing, looking at the octopus and how it processes its visual environment. So in order to interact with our environments, we rely on all of our different senses. whether we are observing people dancing or engaging in solo activities, or maybe we're participating in a group dynamic like playing sports. In all of these cases, we would be using, we receive sensory input, which is like anything we see, hear, smell, feel. All of that needs to be processed within our brain and integrated somehow. And that allows us to generate some sort of perception, which can motivate how we behave in reaction to that. And what I'm most interested in is this part of this loop, how we're processing information and integrating that. Within our brain, we have different regions that are responsible for carrying out these different senses. And we know that different brain regions can both communicate across this wide range and also within their own region. The cells that are driving this communication within a specific region are composed of different cell types, and these form what we know as neuronal circuits. But what we've been kind of aiming to address over years and years of research as more tools become available is how we actually define a cell type. So maybe this is based on shape. So you might notice that some of these are shaped differently than others. This is also influenced by the connections that they make. So you could see here that these neurons are touching and communicating. And then all of these will drive the way that these circuits function. But underlying all of this is gene expression, so molecular signatures that are driving the shape, connections, and function. So within each of the cells in an organism, we have the DNA that's found in the nucleus, and all of the cells have the same exact DNA. So we could think about DNA as providing this insight into a cell's potential to take on a specific identity. But to know what the cell actually becomes, we want to look at the RNA that is transcribed from the DNA. So this will tell us what genes are activated at a certain point. And you can see just in this example of two different cells, these started from the same cell that had equal potential to become a nerve cell or a red blood cell. But based on the cells that, or based on the genes they expressed, they turned into one or the other. By looking at the genes that are activated, those are the ones that will then be translated into proteins, and proteins can tell us about how a cell functions. So a neuron, for example, that might be excited or activated in response to Jennifer Aniston might be what we know as a Jennifer Aniston cell, which is something that they have found in neuroscience. So going back to this loop about how information is processed to generate behavior, I wanted to focus in on this, and specifically during my dissertation, start to understand how these cell types form and how they function in circuits. Normally, people will study this in model organisms. Model organisms will include zebrafish, Drosophila or fruit flies, mice, or even C. elegans, worms. That's because all of these organisms have a short lifespan. They produce tons of offspring, and most importantly, they all have genetic tools that allow us to understand what genes are turned on, what happens if we turn them off, how does it affect the behavior. But what I want to share with you all today is how we can leverage non-model organisms to also learn about neural circuits. So by using non-model organisms, we can begin to learn about evolutionary influences, but we could also learn about fundamental principles. For example, if we look at something that's found in humans and something completely different, and those are the same, something is telling us that that is important for life, for complexity. And what we've been doing here at U of O in the lab that I'm in is using octopus vision as the model for understanding complex brains. We use octopus vision as this model because octopuses are highly visual. So they use their vision for navigating underwater, for engaging in predator avoidance or prey capture, like in this video. And they also rely on their vision for rapid body patterning known as camouflage. Even though we are very, very different from octopuses, we evolved camera type eyes like octopuses, which just means that we have light that focuses through a lens onto a set of cells at the back of the eye. And we did this completely independently. Even though this is similar across humans and octopuses, we know that our brains are very, very different. So as an example, this is what the human brain would look like on the right-hand side, and the octopus brain looks very, very alien-like. But somehow we both are able to process our visual scenes with high resolution and using this camera type eye. So the main question for my dissertation is, how does an octopus process its visual environment, and what can this tell us about how to build a complex brain? Focusing in on the octopus visual system, we could see that it is comprised of both a retina and the optic lobes. So the retina is where I have pointed out at the eye, and the optic lobes are found right behind the eyes, which is actually where the brain is located. So that big little balloon-like structure is their mantle, and that actually is where all of their internal organs and their stomach is, and their brain is found right behind their eyes. In the 1960s, there were studies looking at how these cells are shaped within the visual system. But since then, we've been very limited in technology to understand anything beyond just the way that these cells are shaped. If we looked at a schematic of both the human retina, the eye, and then the octopus visual system, there are also a lot of similarities just based on the way that these cells are shaped. And it seems like even the way that the cells might be communicating, so how close they are to one another, it looks like there are similarities there. But with the technology that was available, they found that photoreceptor cells in octopuses, if you flash a light, these cells become activated. But in humans, if you flash a light, these same cells become deactivated. So even though they look similar, there are some differences in the way that they function. What you probably noticed, though, is in the octopus retina, we really only have like one type of cell. So what we're mostly interested in is what's going on in the optic lobe within this circuit that seems more complex and resembles the other cell types that we see in the human retina. So what we wanted to specifically address with my project is, what are the molecular signatures underlying these cell types? In other words, what are the genes that are expressed in these cells that are driving them to become diverse cell types and form this complex circuit? By addressing this, we would gain insight into these fundamental principles, like what I mentioned, where if there are similarities, it means something is happening with these genes and they're important for high resolution vision. But if they're completely different, that gives us insight into alternative ways of building an advanced visual processing system. This project took very many years to pursue, and we used two main methods that I'm going to try to break down and kind of highlight the main takeaways from our work. So first, to identify the genes that are underlying these diverse cell types, we used single cell RNA sequencing. So that meant that we just took the sequence that is found in these cells, but instead of looking at DNA sequence, which should be the same across all of them, we looked at their RNA to see what's expressed. And then we used this method called fluorescence in situ hybridization, or FISH. And all that means is that we were looking at where these genes are expressed within the tissue. So using some molecular biology and looking at these slices of tissue under a microscope to see how things might be organized. We worked with the octopus bimaculoides. They are about 4 to 6 weeks old when we work with them. And even though a lot of people assume that the octopus is very, very large, we actually work with really small ones. So you can see the speed for scale, for reference to scale. So to do single cell RNA sequencing, we basically will take the optic lobe, since that's the part that we're interested in. We dissociate them into individual cells. That way we can sequence them. And then we'll use this platform where we can sequence the cells, and that will give us what we call a gene expression profile. So this purple cell, cell 1, you can see like for genes A, B, C, it has a different profile compared to genes A, B, C for cell 2, this pink cell. And then once we have all of these information, we're able to then use bioinformatics or computational programs to organize it in a way that we can interpret the relationship between these different cells. These are represented in what we call a UMAP, where each of these dots represents one of the cells that we're looking at. And the distance between the cells represents how similar or dissimilar their gene expression profiles are. And here the color represents the cell class or the cell type that we identified. Since we're interested in neural function, we were interested in looking at neurotransmitter-related genes, because that would give us insight into how the cells might be functioning. So we could look at genes that are related to transportation, to packaging the genes, and the ones that we were mostly interested in are the ones that we know to be involved in vision in humans or other vertebrates. So there's dopamine, glutamate, and acetylcholine, which all have different functions within the visual system. And by looking at different vesicles or transporters related to these genes, so blue would be dopamine, yellow is glutamate, and acetylcholine in orange, we were able to identify different populations within this UMAP that represent different cell types. And in total, we were able to identify 6 major neuronal classes, plus a large population of immature neurons. Once we were able to identify these gene expression profiles and cell types, we wanted to then tie it back to the anatomy and understand where these genes are expressed. So we could then use this FISH technique to look at genes of interest and then stain within the tissue. And if you took a cross-section of the tissue, you can see that there are clear layers that would help us understand how something might be organized. So looking at the example with dopamine, glutamate, and acetylcholine, when we stain for those, you can see that they're expressed in different layers. With dopamine and glutamate, this green and magenta kind of expressed in outer layers and in the deep layer. And acetylcholine is expressed really throughout the entire optic lobe, except for that very outermost layer. So we could then look at all of the individual clusters or groups that make up the cell classes, the different colors, and we identified that there's a lot of diversity within each of these populations. So we systematically went through and looked at all of the gene staining patterns for all of these, and we were able to see that the data within this UMAP correlated with what we were seeing in the anatomy, that there's diversity in the UMAP, and we see this diversity in gene expression as well. What was also very interesting is this large population of immature neurons, which are basically cells that have decided they want to be a neuron, but they haven't really decided what type of cell class or what color they want to be a part of yet. And what was interesting is that a large proportion of these were identified by a novel octopus-specific gene that we discovered, both within this class and many of the other neuronal subgroups as well. So altogether, we've been able to use these 2 big techniques to tie together and put together this molecular cell atlas of the optic lobe, where we reveal both diversity of cell types and then sublaminar organization, so organization within each of the sublayers based on their gene expression. All of these data was building upon what we knew from the 1960s, where there are different cell types based on the way that they're shaped. But by looking at gene expression, we even found more diversity within these different shaped cells. And we also found, compared to the vertebrate retina, that there are some similarities. So you can use this technique to identify more diversity within cell types. There are similar markers that are used that are important for visual processing, but there are also a lot of differences. For example, there are unknown markers that are specific to octopuses, and there's a large population of these immature neurons that we don't really know very much about. And putting this together, we've basically generated a parts list for building a complex visual system. And once genetics are available in octopus, like being able to manipulate genes, we are able to put this out as a resource for people to use. That way, we've kind of saved them time in the beginning. And two ongoing projects right now that start to tie together what the genes are expressed and how this relates to function. One is led by a postdoc and looks at how these different cell types are functioning. And then one project that I'm working on is looking at how different genes are expressed all across the brain outside of just the optic lobe. With that, I want to thank the lab and different collaborators that we've had, as well as the different cores available at U of O. I also want to thank Karen and Larry and the ARCS Foundation and all of you for inviting me. And I've left my contact info in case anyone has questions that they'd like to bring up outside of this talk, in case there's not enough time.
Thank you, May. That was so fascinating. And right off, there's a question that I want to ask you, because I had the question too, and I'm figuring if Chris had it and I had it, there's maybe a few more. When you mentioned building a complex brain, did you mean that literally or figuratively? I mean, what do you mean by building a brain versus understanding how it works?
Yeah. I would say it's more the latter. I know that some research is starting to go into like organoids and actually building these different organs like in a dish. But with something as different as an octopus, we really just want to understand what is required to build or put together the different circuits within their brain.
Got it. Okay, so kind of figuratively, how does this thing work exactly? And then back to Jessie, a couple questions. Jessie, at what depth are these eddies found that you talked to? Or are they all, all over the place?
Yeah, that's a great question. It depends on the eddy, but as from those images that I showed, we were observing them from the surface, but they can actually extend quite deep, and that depends on how old the eddy is, so when it formed and how long it's been, traveling across the ocean. So at the time that we observed them, they seem to maybe be only influencing the top couple hundred meters of the water column, but it's been found that they can actually influence all the way down to that seafloor down at 5,000 meters after a period of time. So that is a good question, and it definitely depends. I think often research on these features observes them at the surface and what's happening in that really shallow layer of the water, but they can definitely extend all the way to the seafloor.
Well, now that brings up another question, and that is, what's the lifespan of an eddy? I mean,
that's another great question. It also depends, I think, on a lot of different factors. The ones that I talked about in the Eastern Pacific have lifespans of about 6 months. So they spin up, sort of off the coast of Central America, and they propagate westward, and they usually fizzle out you know, probably about halfway, you know, towards Hawaii, and then it'll take them about 6 months to get there. So in other areas, they can have much shorter lifespans or slightly longer lifespans. It just depends a little bit on the general ocean dynamics in a given region.
And then another question for you now that we've got you on the hot seat. This one's from your colleague, May.
Okay.
So does the temperature affect the day-night migration cycle at all?
It does, yeah. I would say it doesn't influence whether or not it happens, because that's generally driven by light. And so the species that are going to do this migration are always going to do it. But part of the research that I did looked into different factors, like how much does temperature influence maybe how many animals are migrating or how deep they migrate, or perhaps how long they stay at the surface before going back down. So temperature can definitely have an influence. As I mentioned, oxygen can have an influence. Light has a big influence. So we've tried to look at the different factors that affect, you know, what the structure, the different behaviors of that migration could be.
And an early question during your presentation was, do the organisms that go to the surface in this vertical migration at night, do they see, or are they using other senses to detect where they are and where they want to be?
Largely, most of these animals can see, because during the day they're in that twilight zone area. There's enough light that they're actually often quite specially adapted to seeing in low light conditions. But interestingly, animals that live a lot deeper in the water column might not be able to see very well because there's no light and they have little use for eyes. So these animals can largely see. They definitely rely on other senses. A lot of fish, for example, have these receptors on their bodies to be able to detect pressure differences, to feel if something might swim by, if there's a wave, a pressure wave in the water nearby them. So they'll often use senses like that, or senses of smell, which is often used for communication with other organisms. But vision is definitely really important because a number of these animals have bioluminescence, so they might have these little lights on their bodies that can let other animals of the same species know that they're there or that they are the same species. It might help them find mates or just find others of their own species. So to that question, yes, a lot of these animals can see.
Excellent. Are there any other questions from anyone? Let's see, it looks like there might be one here. May, this one's for May. Are you familiar with the 2020 documentary, My Octopus Teacher? Oh my goodness, that was great. It's a wonderful testament to the intelligence of this beloved mollusk.
Yeah, I did watch that, and I did find that it reflected a lot of what we see, even just with the smaller animals that we work with.
And then one for Jessica. Are you aware that one of your collaborators, Jeff Drazen, is also an ARCS Scholar alum? Yeah, she knew that.
I was aware, yes. He's actually my PhD advisor, so very small world.
Oh, that's great. Here's another question for you, Jessie. In the mining license process, is the mining license process paying attention to the research that you and your colleagues are doing, and if or how it can be done without very bad consequences?
Yeah, it is paying attention. There's actually requirements for these different contractors to conduct environmental baseline surveys, which is part of the research that we conducted was to fulfill that requirement of theirs, and to really go out and be the first research happening in this domain. So I would say they're paying attention to it. I think, how the information is used, obviously there's conflicting interests of companies that want to go mine, but obviously stewards that want to mitigate any potential damage. I will say it is a unique scenario where there is the opportunity to go out and take this precautionary approach and do all this research before this industry spins up, because that's obviously not the case with other industries like drilling for oil, for example, in the Gulf of Mexico. I think when big disasters happen, there wasn't necessarily all that baseline research behind it. So it is a unique opportunity, and we really hope that that the information that we are relaying to these mining agencies is going to be used, and in some capacity it is mandated to be used. So.
And for both of you, at what point did you realize that you had an interest in science, and how did you get into your respective fields? May, you want to go first?
Sure. I was very intimidated by science really for most of my life. But in undergrad, I was already pursuing psychology because I was curious about the brain, and then just learning about different routes you can take. I was told that you would have more financial stability if you pursued a PhD, whatever that is. So that's kind of how I started getting involved in research, just working towards that goal. And then I ended up working with octopus, I think just by chance. I was initially working with humans, but when I was interviewing at different places, I saw the octopus and learned about how weird their brain is, and something about that was just very appealing, kind of challenging, but like also very interesting to pursue.
Thank you so much. Jessie, what was your path?
Yeah, I maybe didn't have a straightforward path so much, but I was always interested in biology, and knew I wanted to study this in college, and really had an interest actually in potentially going to veterinary medicine and studying marine animals from a medical sense. And as I got more into just general marine biology and ocean research, quickly realized how interesting I found that work, and how much I wanted to pursue research more than medical sciences. And so through just different opportunities to conduct research in different lab spaces and go do field work in different parts of the world, I think that just really led to a developing passion for doing ocean research, and particularly in parts of the ocean where people don't often think of or go. I grew up nowhere near the ocean, so the idea that I can, go out into the middle of the ocean and build a career off of that was really inspiring. So.
Well, we're very glad you both found your way into these areas that are so important, and we're so proud of you, proud to call you ARCS scholars. Thank you for all you're doing, and we're certainly going to be watching you going forward. Thank you so much for being part of today's program, Jessie and May. Your work, as you can tell from all the interest here in the chat, everyone's clapping, so important, so interesting. We're so grateful to you for giving back to ARCS by sharing your research and career highlights with us today. Thank you all for participating in the session. We'll send out a link to the recording for you to share. You will also receive a short survey. Please let us know if there's a topic of interest to you. We really do use the survey information to design our sessions, so please return them. The next ARCS Forward event will be September 20th, 2023 at noon Eastern Time, 9 o'clock Coast Time, and all of you assort yourselves in between accordingly. We are very excited to feature our newest ARCS Alumni Hall of Fame inductee, Dr. Heidi Hamill. Dr. Hamill will be talking with Dr. Ori Fox about their work in planetary astronomy. So from the depths of the ocean to the arc overhead, ARCS can bring it. Thank you for attending. We look forward to seeing you in September. Thanks, everyone. Have a great summer.
Thank you.
Thank you, everyone.
Great job.